Hello everyone, welcome back to our new session. Today we are going to discuss about the cell wall, plasma membrane and extracellular matrix. Myself, Sri Lakshmi, 2nd MSc Biology from St. Mary's College, Thrissur. Cell wall. Cell wall is a structural layer that surrounds some type of cells or tissues which is situated outside the cell membrane. Cell wall was first observed and named simply as wall by Robert Hooke in 1665. In 1804, the Carl Rudolphi and J.H.F. Link proved that cells have an independent cell wall. It is tough, flexible, and rigid, also provide the cell both structural support and protection. In case of animals, they do not have cell wall. It mainly present in plants, fungi, bacteria, algae, and some type of archaea. On the basis of chemical composition, there are three types of cell wall: bacterial cell wall of fungi, and plant cell wall. In case of bacteria, it is mainly made up of mucopeptides and muramic acid. In case of fungi, it is made up of chitin. And in case of plants, it is mainly made up of cellulose and hemicellulose microfibrils. So, first we can tell about the plant cell wall. Actually, plant cell wall is a multi-layered and it consists of three sections: middle lamella, primary cell wall, and secondary cell wall. All the plants contain middle lamella and primary cell wall, but not all have secondary cell wall. Middle lamella is the outermost layer which contain polysaccharides and pectin which aid in cell adhesion. The second layer of plant cell wall is primary cell wall. It is the layer which is formed between the middle lamella and plasma membrane. It is primarily composed of cellulose and hemicellulose microfibrils. Primary cell wall provide the strength and flexibility needed for the cell growth. The third layer is secondary cell wall. It is the layer which is formed between the primary cell wall and plasma membrane. Once the primary cell wall has stopped its dividing and growing, it may thicken to form a secondary cell wall. This rigid layer strengthen and support the cell. In addition to cellulose and hemicellulose, some secondary cell wall contain lignin which also strengthen the cell wall. Functions of plant cell wall. The plant cell wall prevent the enlargement of plants and it play an important role in the absorption, transport and secretion of substances of plants. It provide the protection from physical injury. Together with vacuole, plant cell wall provide skeletal support and also it provide the plant cell a definite shape and size to give the rigidity and strength to the cell. The next type of cell wall is fungi cell wall. We are all know fungi are eukaryotes. It mainly consists of three main components: chitin, glucan, and proteins. Chitins are the polymers that consist mainly of unbranched chain of beta-1,4 linked N-acetyl glucosamine. In the case of Ascomycota and Basidium mycota, it contains poly beta-1,4 linked N-acetyl glucosamine, that is chitosan. Both the chitin and chitosans are synthesized and extruded at the plasma membrane. The second component of fungi cell wall is glucans. These are the glucose polymers that function to cross-link chitin or chitosan polymers. In case of beta glucans, these are the glucose molecule linked via beta-1,3 or 1,6 bond and also provide rigidity to the cell wall. While alpha glucans are defined by alpha-1,3 or alpha-1,4 bond and function as a part of matrix. Third component is proteins. The enzymes necessary for all synthesis of cell wall and lysis in addition to structural proteins are all present in the cell wall. Most of the structural proteins found in the cell wall are glycosylated and contain mannose. Thus, these proteins are called mannose protein or mannans. Functions of fungi cell wall. They protect against the osmotic lysis, act as a molecular sieve, and they contain pigments for the protection, act as a binding site for enzymes. The outermost surface of fungi cell wall provide the medium between the cell and the environment. The third type of cell wall is bacterial cell wall. Bacterial cell walls are made up of peptidoglycan, which is also known as murin. These are made from polysaccharide chains cross-linked by unusual peptides containing D amino acids. Peptidoglycan or murin is a polymer consisting of sugar and amino acids that form a mesh-like layer outside the cell membrane. The sugar components consist of alternating residues of beta-1,4 linked N-acetylglucosamine and N-acetylmuramic acid. On the basis of cell wall structure and their response to strain, bacteria have been classified into two types: gram-positive bacteria and gram-negative bacteria. In case of gram-positive cell wall, usually they are thick and homogeneous. It is mainly composed of peptidoglycans. It contains large amount of teichoic acid. But in case of gram-negative cell wall, they are multi-layered and more complex than gram-positive cell wall. The peptidoglycan it contains is very thin. Most abundant protein in gram-negative cell wall is brownish lipoprotein. Functions of bacterial cell wall. They protect the cell against osmotic shock and physical damage. Regulation of substances transport into and out of the cells. Help in the movement of cells and also help in the protein synthesis. They act as a mineral storage of cells. It contains supplemental genetic information such as resistance to antibiotics, production of toxins, and tolerance to toxic environment. Next, we can discuss about the extracellular matrix. Actually, it is a three-dimensional network of extracellular macromolecules such as collagen, glycoprotein, and enzymes. They provide the structural and biochemical support to the surrounding cells. It is mainly abbreviated as ECM. The living tissues are not just accumulation of tightly packed cells. Much of a tissue's volume is made up of extracellular space, and this void is filled with a complex meshwork called extracellular matrix. Mainly, ECM is made up of proteoglycan, water, minerals, and fibrous protein. Proteoglycan is made up of glycosaminoglycan, that is a protein core surrounded by long chain of starch-like molecule. The two main classes of molecule that can be found in the ECM is fibrous protein and proteoglycan. First one is fibrous protein. There are several type of fibrous protein which is include collagen, elastin, fibronectin and laminin. These fibrous proteins are mainly produced by the fibroblast. Collagen is a strong, stretch resistant fiber which provides tensile strength to tissues. It is the most abundant protein in the human body and also the principal constituent of ligaments which provide support to the skin. Elastin is a stretchy and resilient protein. Much like rubber band, they permit the tissue to return to their original shape after they have been stretched. In case of laminin, they form a sheet-like network that serves as a glue between the dissimilar tissues. It is the principal protein in the basement protein and also present in the connective tissues. Fibronectin is a protein which is secreted from the fibroblast in a water soluble form, but it is quickly assembled into an insoluble meshwork. It is mainly important during the embryonic development, also necessary for the cellular division and specialization in many tissues. The second class of is proteoglycan. In contrast to fibrous protein, it provides resistance to stretching forces. Mainly, it provides resistance to the compressive or squashing forces. Functions of ECM. It provides both the structural and biochemical support. Help in the segregation of tissue from one another. Help to regulate the intercellular communication and also regulate the cell dynamic behavior. The third and last topic of our The third and last topic of our section is plasma membrane. Plasma membrane is also known as plasma lemma. It is an elastic porous, selectively or differentially permeable and biologically active living membrane. It has a lipoprotein composition and lamellar organization and semi-fluid consistency. Mainly, the plasma membrane is formed of water, proteins, lipids, and carbohydrates. Water molecules are seen on the inner and outer boundaries. The lipid and protein form the non-aqueous inner core. But in case of carbohydrates, they are seen on the external surface of plasma membrane, mostly serving as a cell coat. Lipids form the structural framework of the membrane, while proteins have both the structural and functional roles. Structural protein link with the lipid forming lipoprotein, while the functional proteins serve as enzymes, antigens, carrier molecules, and receptor molecules. The carbohydrate link with protein and lipid, forming glycoprotein and glycolipid respectively. They give additional strength and stability to the lipoprotein framework. Plasma membrane has a lamellar or micellar organization. In laminar organization, the molecules are arranged in thin sheets or layer. But in micellar organization, they are arranged as globular subunits called micelles. There are several theories have been advanced to explain the molecular organization of plasma membrane. They are trilamellar concepts of Danieli and Davison, unit membrane concept of Robertson, and fluid mosaic model of Singer and Nicholson. Functions of plasma membrane. They serve as a selective gateway and regulate the inflow and outflow of materials, help in the recognition of foreign cells. They maintain the osmotic balance between the intracellular cytoplasm and extracellular fluids. They maintain the ionic equilibrium between the cytoplasm and surrounding medium. Take the useful substances by endocytosis and active and passive mechanisms and give out the waste material or cell products by exocytosis. Thank you.